Alright, so now I'm going to switch to a video that really talks about DNA replication as a whole, and then we'll talk about this together. Here we're going to talk about bacteria cell division. And so here we have some E. coli that we're going to see in this dish. And in order to divide, all cells, bacteria and eukaryotic cells, need to grow first. Then they need to copy the DNA. And then they can split, and now each cell has a full copy of the genomic DNA. So here we see some purple bacteria. They're growing, then they will split by fission, but before they split, they have to copy the genome so that both of these cells have a full copy. And here we see DNA with our double helix that we recognize by now. And we know that the phosphodiester linkages link the nucleotides of a single strand together, and then the other strand also linked by phosphodiester linkages. And then the bases, the nucleotide bases, stick into the middle, and they're held together by hydrogen bonds. So this is the chemistry, and we can see a single nucleotide here with its phosphate, ribose sugar, and base. Right? And we have the A, T, C, and G as nitrogenous bases, and we know that the phosphodiester linkage is by ribose and phosphate next to the next ribose and phosphate. And thymine binds to A, T binds to A with 2 hydrogen bonds, and C binds to G with 3 hydrogen bonds sticking in the middle. And that's what holds our 2 anti-parallel strands together. And we also know that we have carbon 1, 2, 3, 4, and 5, which is why we have a 5' end and a 3' end, and our nucleotide strands go anti-parallel, so if one is 5 to 3, the other is going to be 3 to 5. So let's take a quick look at this and make sure we can see this. So we know that the phosphate group is linked to the 5' carbon, that carbon that sits out here outside of the ribose ring. So this would be carbon 1 here, carbon 2, carbon 3, carbon 4, and carbon 5. So this is the 5' end of this nucleotide, and then the strand ends at the 3' end down here. Conversely, this would be the 3' end, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So the anti-parallel strand is 3' down here to 5'. So they're in opposite directions. And we know that G always binds to C with its 3 hydrogen bonds, T always binds to A with its 2 hydrogen bonds, and that is the complementary base pairing right there.
This shows the anti-parallel nature of the DNA strands.
Alright, so let's think about this a little bit more. So the part that we're going to learn about next is the fact that this base pairing, this complementary base pairing, A always binding to T, G always binding to C, allows for replication. This is how it can act as its own template. That's another word you're going to hear about a lot. All right? So the beauty of the structure of DNA explains how the genetic material can contain information and how it can be replicated. Right? And this is what pretty much this entire chapter is going to be about, that second point there. It also explains how the genetic material can contain different varieties of the same genes. So really it shows how the sequence of DNA can code for different proteins. And this is the basis for chapter 10, which we'll start next week. Right? But here we're really going to focus on DNA replication and how the fact that A binds to T, G binds to C, how that facilitates DNA replication. All right, there are 4 crucial steps in division for any type of cell. Cell growth, copy the genome, separate the chromosomes, and divide. All right, so why does the cell need to grow before it divides? What would happen if that wasn't the case? Well, if that wasn't the case, then the next cells would both be half the size. And if those didn't grow before they divided, the next generation would be a quarter of the size, and so forth. So if you think about it with your own cell division, if your cell, when you were just a fertilized egg, that single cell, now you are 10 trillion cells. If your cells didn't grow before they divided so that the new cells are roughly the same size as the original one was, then you would basically be 10 trillion cells but still the same size as that original fertilized egg. So your cells have to grow before they divide. All right? And within that growth is also kind of making sure your mitochondria, all the various organelles, at least for eukaryotic cells that is, not bacteria, but making sure those organelles divide and get distributed evenly as well. Why does the genome need to be copied before we have two cells? Well, if the genome is not copied, then your two cells, basically, one of them, it would either have no genetic information, and that would cause it to die, or both of them would end up half of the genetic information. I realize that now that we are remote at the moment, this analogy doesn't make quite as much sense, but normally I would give you your exams on paper. And so when I write the exam, what do I need to do in order to be able to give all 40 some odd of you a copy of the exam? Well, I need to photocopy it. But when I photocopy it, I need to photocopy the whole thing. Every one of you needs that entire exam. Every one of these cells needs the entire genome, or they're not going to have all the information needed to make that cell. So you need the full copy of the genome. Otherwise, it would be like me taking, if, let's say this is your exam, and in order to make multiple copies, I just rip it in half and give, okay, there's 2 copies now. Oh, now there's another set of copies. Well, that wouldn't work because each of you are getting just a small portion of that exam. And while you might like the fact that you're not taking as much of the exam, your grade would suffer because you're not getting full credit for the whole exam. Here, they need the full genome. Each cell needs the full genome. So every time a cell divides, that genome has to be copied so that both cells, each cell, can get a full copy. Right? So basically you make 2 copies right before that cell divides, then you send 1 copy to each cell. And for human cells, the genome is 46 chromosomes. So all 46 of those chromosomes have to be copied, and then you have to make sure that each cell gets the right combination. Right? So if we have 2 chromosome 1s, one from mom, one from dad. We'll learn all about this in the genetics. Well, this cell needs 2 of chromosome 1, and this cell needs 2 of chromosome 1. It doesn't work for this cell to get 4 of chromosome 1 and none of chromosome 2, and vice versa. So you need to have equal copies. So you copy the 46 chromosomes, and then you send each of the 46 chromosomes to the 2 different cells. Right? And then finally, once that happens, the cell is ready to divide. So this is what has to happen for the cell to divide. You need to grow, you need to copy the genome, and then you need to separate the chromosome so that each new cell gets the full set complement of genes, the full genome. Then the cell can divide. All right, so how does this DNA copying happen? So here is a quote from Watson and Crick, 1953. "It has not escaped our attention that the specific base pairing we have postulated immediately suggests a copying mechanism for the genetic material." Well, what are we talking about here? Right? We're going to get to this in a second, but the whole basis of being able to copy the genetic material is that A always binds to T for DNA, and G always binds to C. If it's RNA, A will bind to U, but that's kind of the same idea as A always binding to T. And here we're talking about DNA, so A binds to T, 2 hydrogen bonds, G binds to C with 3 hydrogen bonds.